All right, so I apologize. This is a few days late. I just kind of got everything back up and running on my computer and the recording software working appropriately. So I know most of you may not have time to view this, but we will next talk about plant reproduction and then animal reproduction. This is just kind of an overall view of cell cycles and cellular division. We will talk about both meiosis and mitosis, and we will talk about the different phases of them and give you some general overviews about each of them. So when we're talking about cell division, in order to reproduce, all cells have to go through this process. And cellular reproduction is taking place all the time within your body. Cells are constantly reproducing. Plant cells out there are constantly reproducing. All living organisms have to go through cellular division throughout their life. It doesn't just have to do with reproduction and producing offspring. And this actual process varies depending on the cell type. So we're going to talk about some different types of cells that we have and some of the things that happen with it. So first we have somatic cells. These are non-reproductive cells and they reproduce by mitosis. So these are not used for reproduction purposes. They're just cells that are used in order to be able to make new cells and through that cellular division. Then we have gametic cellular division, and these are producing gametes, and these are used for reproduction. So they go through mitosis, and then they go through meiosis. Now, if you remember, we had a couple of different cell types. We had prokaryotes and eukaryotes. Well, we have our prokaryotes that are going to go through somatic cell division, even for their reproduction. So that's just one little thing to kind of point out and keep a little different in your mind, because they do not, whenever bacteria reproduce, they don't have any gamete cells because they are just producing exact copies of themselves. So that's one way to think about somatic cell division, is the cell is actually just making an exact copy of itself through mitosis. We have gametic cell division, which the cells can be making a copy of themselves if it's asexual reproduction, or if it's sexual reproduction, there's going to be other processes involved. But they utilize the first part of mitosis, and then they go through meiosis, which is a second part of the cell division and joining together of cells from two different parents. So when we look at the cell cycle, most of the lifespan of the cell is spent in what we call interphase. And interphase is the time between cell divisions. So interphase is where the cell is doing a lot of the prep work, if you will, in between cellular division. So as we kind of look at how this is going to take place, the first thing is during interphase, this is what takes place. First of all, the DNA takes the form of chromatin. And when we're talking about taking the form of chromatin, this is where it's a long strand of DNA wrapped around a cluster of protein called a histone. This, again, is only in plants and animals and eukaryotic organisms that this happens. And you can see these chromatin right here wrapped around these histones that are present right here that look like little spheres. And you can see here the chromatin and the histones that are all packed in here. So we have we have this chromatin and we have these long strands of DNA. Well, what happens is during mitosis, the chromatin condenses into a chromosome. So you can see here we kind of have this organization taking place. We're small. We get a little larger when we attach to the chromatin. We bundle several of the chromatins together. We now make an entire chain in an extended form. So we're starting to kind of look like a section of a chromosome. Now we condense that section of chromosome here. And what we end up with is the entire mitotic chromosome. So we have an entire mitotic chromosome down here. And you can see how we have the two sister chromatids, or we have the pair that make up the chromosome here. So we know we're dealing with eukaryotic organisms because we have the linear pairs of chromosomes. So this is, if you're looking here at human cells, Human cells have 46 chromosomes. This includes 23 from the mother and 23 from the father. You have 22 pairs of chromosomes that carry traits, and then you have one pair of sex chromosomes that will distinguish or determine whether the offspring is male or female. So what we're looking at here is, again, during interphase. And you can look, we won't be looking at these under the microscope in lab because you can see it right here. We can see pictures of it that show it much better. But this is interphase, and it can be identified under the microscope. And you can look for the nuclei that contain the chromatin, as opposed to a more condensed worm-like chromosome. So what we're looking for is we're looking for the chromatin, and those chromatin kind of look like a nuclei. So you can see here, this looks like a nuclei inside the cell. You can see all these cells that have the chromatin that looks like a nuclei, and they don't look like the worm structure here. So this is going through some form of mitosis. This is still going through some form of mitosis as well. But you can see the condensed form here, and we have the actual nuclei. The nucleolus may be as visible as a dark area. So you can see the nucleolus in here. The nucleolus is the area with the genetic material or the chromosomes inside of the nucleus. So you can see the condensed dark area even more so in here versus the lighter area of the nucleus outside. So now what we're doing is we're looking through these processes. This is the entire cell cycle over here in this circle on the left, and that's what we're going to be working through here. So interphase consists of 3 different phases. So when we're looking at interphase here, you can see we're looking at interphase. So we have these 3 distinct phases that are in blue, the G1, the S, and the G2. During the G1 phase, and by the way, this is on your worksheet and do expect it on the test. During the G1 phase, the cell replicates its organelles and cytosol so that there is enough for 2 functional cells. So we replicate itself, and it's basically replication or duplication, as you see here. So the normal cell functions, the cell is growing, but we're duplicating organelles, we're doing all that stuff. So we're still functioning, but we are creating a copy of the cell. During the S phase, the DNA of the cell is completely duplicated. So that's what takes place here, complete duplication of the DNA during the S phase. Finally, to the G2 phase, this is additional growth is needed so the cell can grow some more, can prepare, and protein synthesis is going to take place before we get into the actual cell division. So think of the interphase here as kind of ramping up the procedures and ready to go. You can see it takes 8 hours, 5 hours if you want to go that route, 21 hours. So almost a full 24 hours here but in interphase. And remember, we said it spends the most time there. And if you look at this, it spends 8 times more time in interphase than it does down here in cytokinesis. So we all love donuts. So this is what we're going to look at as an example. And I wish I had the ability to turn 1 donut into 2. I'd be even fatter than I am. But mitosis is the part of the cell cycle where the nucleus physically divides into 2, each containing an exact copy of the original DNA. Excuse me, I had to get a drink. But remember, mitosis, we are just splitting the cell essentially and creating an exact copy. So you can see what we start out with here, and you can see it going through the process. You can see the chromosomes kind of aligning here, moving toward the poles on our spindle fibers, pulling apart, then rearranging inside, starting to rearrange in our new chromatins. And then you look here, and we have an exact replica of what we had when we began. Except we just have 2 of them now, where we had 1 to begin. So now we're going to look at that cytokinesis part. So we're actually looking at the forms of mitosis now and the stages of it. So during prophase, the DNA begins to condense and form chromosomes. It's highlighting it here. Okay, so we're starting to get the condensing here and forming the chromosomes. The nucleolus disappears. So see, there's no dark area in here. Like we can see a dark area here. You can see some dark areas up here. You don't see that in the one that we are highlighting. The spindle fibers are formed by the centrioles and they attach to the centromeres. Now, if we jump back here, these would be the spindle fibers, would be these pink colored lines here. The centrioles would be out here, and these would be the centromeres in the middle. Okay? So then the two are joined. There are two joined copies of the chromosome before mitosis called chromatids. So we then have those two chromatids that are a copy of each chromosome. The chromatids are connected by a single point called the centromere. And as you look here, this is the centromere right here that's connecting these chromatids. You can see how they're connected in there. And then you can see the other parts of it. The whole thing is a chromatid, but the centromere connects them. So that was the first phase. That's the prophase phase of mitosis. We then move to metaphase. Metaphase is where the chromosomes move and align themselves in a narrow central zone in the cell. So they kind of start to migrate toward the center of the cell. And this is kind of what it's going to look like. See how they've migrated toward the center? They're in the center. These would be the poles. And as we have this migration, they're in the center of the cell during metaphase. And what happens is the chromosome don't move independently. They have to migrate along these microtubules. And these microtubules are called spindles or spindle fibers. And these microtubules are what hold the chromosomes in place. And you can see how they're aligned along these spindles if you look through here. So now what we have is we have anaphase. Anaphase is the third stage of mitosis, and it begins when the centromere of each chromatid splits. Now, let's go back and let's look at where it splits. When they split along the they split along the centromere. So I want to be clear about that as to where they split. They split along that centromere. So now we have daughter chromosomes, because these daughter chromosomes are going to be identical in nature. Okay? They are identical to one another. And they are pulled toward opposite ends of the spindle apparatus. So now you can see where we are. They've been pulled toward the center. And you can see what's happened here. When the chromosome is now fully divided, it forms two nuclear membranes during what we have as the final stage of mitosis, which is telophase. The DNA is returned to its uncoiled chromatin form, and the nucleolus becomes visible again. So this cell essentially has split into two identical copies now. So cytokinesis is the process of mitosis only. The cytosol and the organelles in the cell membrane are physically divided during cytokinesis. The cells return to the interphase point. So if we go back in time here, and I know I'm going fast and confusing, but now we have completed interphase, we've completed cytokinesis. Notice that after telophase, we return to interphase again. I'm going to jump back forward to where we need to go. All right. So now that we have jumped back forward, we're going to talk about cytokinesis and what is happening. Okay, cytokinesis produces and proceeds different in plant cells and animal cells. There's different processes that take place, and there's different names for the processes that take place. Remember, plant cells have a cell wall, animal cells do not. So in animal cells, cytokinesis causes the formation of a shallow indentation called the cleavage furrow. So this is the cleavage furrow that takes place in animal cells. The furrow tightens So think of it as taking a piece of string and putting it around your finger, and you pull it tight, and that's what starts to happen. It eventually would pinch shut. Don't try it at home. You're not going to pull your finger off or pinch it off. I think we all know that. A plant cell, on the other hand, has vesicles that have that cell wall, and these vesicles start to form a cell plate. So remember, this is the difference in having a cell wall and not having a cell wall. So we have a cleavage furrow in plants and animal cells, and we have a cell plate in plant cells. So now we're just going to kind of look through. These are epithelial cells, which would be skin cells of the inside of your cheek or something. This is a time-lapse microscopy of the epithelial cell, and we will actually see if it works. So I'm not sure, but we're going to give it a try. So there was no sound with that, so we're going to kind of talk through this a little bit, and I'm going to pause it as we go along. I know it's 8 seconds, but what we can see, this is going through the process of mitosis. So as we start, we've already been through interphase. Okay, so we are ready for the prophase, and the prophase is where we've already kind of gotten into prophase. We're ready to look at metaphase, where they're lined up in the center here. Okay, see them lined up in the center? So as we start watching this, now you can see where they've lined up in the center. So if we back this up slowly, you can see how they are just arranged. You can see they're in the nucleolus. You can see how they're contained in the nucleolus around here, but you can still see our spindles at the poles. As we begin to advance forward, you can see it start to take shape, but you can see what the chromosomes start to do. They have unwound from their from where they're supposed to, and they will start to form in the middle as we go from prophase into metaphase. So we are now in metaphase. You can see them lined up in the center. And as you move on, now you can see that they're moving along the spindle fibers that you can see in here now. So they're moving along, and what we start to see now is, oh, and it just happened right there. You can see how they start to pull apart. See how they're starting to pull apart? That's anaphase. And they're pulling apart right at that centromere. And if we stop it right there, you can see how they've pulled apart, and we have identical pairs of chromosomes being pulled apart. And we kind of jump forward. You can see them being pulled to the poles. We are now, you can see right there. You can see this is an epithelial cell. It says a pig kidney. So this is where our cleavage furrow is going to be present. Animal cell cleavage furrow. And it comes through and they pull out. See the cleavage furrow pinching it off. And as it pinches off, you have two separate cells. So I'm going to let it play the whole way through one more time, and you can watch it. And there you have it. Pretty cool, huh? All right, so now we're going to get back to our presentation. And you can kind of see this process takes not 8 seconds. It takes about 20 minutes to take place. Because remember, and sometimes some cells can take up to 1 to 3 hours, but never much longer than that. Interphase is where they're going to spend a lot more time. So, as we again look at this cell cycle, once the cell division is complete, the daughter cells will re-enter interphase, signaling that they will divide once again. So we have the cell that's divided, and they'll keep doing it. Those cells will just keep on dividing, and this process replicates over and over. Now, let's talk a little bit about cancer. Most multicellular organisms do not divide constantly. Rather, they have signals or conditions that initiate the process of mitosis. Well, cancer kind of overrides that. And cancer means that we kind of have unprogrammed cellular growth. So with cancer, there's a problem because you have cells that are just being created for cell's sake and they're no real purpose. There are proteins that are secreted by the body. They stimulate the cell to divide and we get cancerous cells. So you can kind of see what happens with a lot of these different things with cancer and with different mammal cells. Cells exhibit different dependencies, and they have to come in contact with solid surfaces. Those cancer cells will disregard all of those controls, and they will then form masses, which are tumors. Now what it talks about is if the tumor is encapsulated by other tissue and doesn't spread, that tumor is said to be benign. However, if the tumor is part and it's able to break free and spread throughout the circulatory system, it can be malignant, and this means that it's present in other places and is able to spread. So that took care of mitosis and that somatic cell division. The next thing we're going to work through is we're going to look through the gametic cell division, which is for sexual reproduction, cellular reproduction. In multicellular organisms, mitosis is primarily used for living tissue regeneration and repair. However, mitosis is the primary form in unicellular organisms like we talked about. In unicellular organisms, mitosis is the primary form of reproduction. So it forms in reproduction in those bacteria and those prokaryotes and single-celled organisms. Even some of our single-celled eukaryotes, for example, protozoans and algae, may go through mitosis only. So now we're going to look at two different types of reproduction. We have asexual reproduction, which involves a single parent of an offspring and a single offspring. The offspring are produced are in most cases genetically identical to the parents. Asexual reproduction is simple, efficient, and effective. It uses only mitosis. Prokaryotes, or our bacteria, all reproduce asexually. There are a few organisms like sponges that reproduce asexually as well. Sexual reproduction, the offspring are produced by the fusion of two sex cells, one from each parent. These fuse into a single cell before the offspring can grow. The offspring must inherit genetic information from each parent. They get a split. Most animals and plants and some single-celled organisms will reproduce sexually. Organisms that reproduce sexually do not produce a clone. That means they have genetic diversity within their offspring. This requires the use of mitosis and then meiosis. And specifically later when we look at animal reproduction, we'll look at the somatic division of the sperm and the egg and then the joining together of the two of them to get through meiosis. So we're going to look at spermatogenesis here first. The male gametes are called sperm, and these begin as stem cells in the testes. During spermatogenesis, the formation of sperm, which is the stem cells, they undergo mitosis to produce two exact copies. So spermatogenesis is where we just make two exact copies of the sperm of the stem cells. And we have then two daughter cells of sperm in the testes. Only one copy remains behind. Sorry for that brief delay. I had a phone call. But we left off with spermatogenesis. And we know that spermatogenesis, one copy remains behind to generate future sperm, and the other copy migrates toward the epididymis to be used in reproduction. So this is kind of what it looks like. At this point, the gametes, because remember we're going through gametogenesis here, the gametes are given the diploid or the 2N notation, meaning they contain a full set of 46 chromosomes. The daughter cell that migrates away will undergo a second type of cell division called meiosis, which will result in the production of 4 genetically different cells, each having only half the number of normal chromosomes. These are referred to as the haploid. So the haploid is just the N notation, where the diploid has the 2N notation. So meiosis, we're going to look here, meiosis 1, we have meiosis 1 and we have meiosis 2. There are two separate sets of cell division that occur in meiosis. They are labeled meiosis 1 and meiosis 2. We can see kind of some of what they do and take place. A lot of the names will be the same, but just understand that there are some differences. During prophase 1, the homologous chromosomes from each of the father's parents come together to form a tetrid. A tetrid means 4. Okay? So we have our tetrid, and they form the tetrid, and they form a tetrid instead of remaining separate. So we have our tetrid formed. Okay? The tetrids then line up in the middle during metaphase 1. So they have lined up in the middle here now. The tetrids don't necessarily have to be in the middle, it just shows it here. But metaphase 1, the tetrids are lined up in the middle. So when we have metaphase 1, right here, lined up in the middle. Prophase 1 would kind of be over here, I guess, whenever we talk about it. But this is kind of prophase 1 and metaphase 1 kind of represented all at once. Now, we'll talk about some of the rest of the things that happen here. We get into anaphase, pulling apart, and then anaphase 1, telophase 1 happening over here. So here's what happens, this phenomenon, which is called crossing over. And this is a unique phenomenon that occurs in the prophase 1 of meiosis. Homologous chromosomes exchange segments and produce combinations of genes that make it unique. This is what produces that genetic diversity. Blue indicates the chromosomes from the father, red is from the mother. And this crossing over is what gives us that different genetic diversity. So we're back here. Following anaphase 1 and telophase 1, so we have anaphase 1 here, telophase 1 here. So in telophase 1 is when we have the 2N, or that diploid number. So the stem cells have divided into 2 haploid daughter cells. Okay? So we're going to kind of look here, and the stages of meiosis 2, which meiosis 2 start right here, and I have another phone call. Hold on. And we're back. I apologize. So the stages of meiosis 2 mirror what happens during mitosis. So we have the beginning here, where we have our 2 haploid daughter cells. Those 2 haploid daughter cells will then duplicate. As they duplicate, we have this. So now we have our haploid, or spermatids. So those spermatids then become actual sperm cells. And remember, the end result is 4 genetically different daughter cells. That's important to remember, that they are genetically different. So as we look through then, that was spermatogenesis, which took place in the male or the sperm. This is going to be oogenesis, which is the ovaries produce eggs. Cytoplasm is concentrated in one of the 4 daughter cells. The rest are what we call polar bodies, and they get reabsorbed. So this looks a little different. So we have the process that takes place. We have over here, this is kind of meiosis 1 and meiosis 2. So you can see we have, during metaphase here, we have prophase, we have our metaphase, we have our anaphase. When we get to telophase, which is where meiosis 1 shifts over to meiosis 2, then during this process, we have our cytoplasm, and we have our daughter cell, but this is what we call like the main daughter cell, and the main daughter cell is what goes through the whole process. They all go through meiosis 2, but only the main one is going to produce and end up being the haploid egg cell out here. So this is how we get the gametes. So we then ended up with the male gamete here and the female gamete over here, or the sperm and the egg. The rest of these are referred to as polar bodies, and they get reabsorbed back into the body. That is all we have for this evening. There will be a study guide that goes up. I hope you get to watch this. We will be talking about it in class tomorrow evening. Thank you, and have a good evening.